Hello, good afternoon, everyone. It's nice to be back. Okay, it's been a long while since I had my last class because it was the holidays and I think everyone needed a break. All right. Okay, so do I. Okay, so everyone had a nice holiday and then now it's back to business. Okay, everyone has started schooling. So I presume by now probably most of the schools or most of the teachers have already done chapter 1 and chapter 2. So I'm sort of like trying to flow with the syllabus. So I'm going to start with chapter 3. Okay, so probably most of you are at chapter 3 or maybe have just going to end chapter 2. Okay, so it's nice to see all of you here. So I'm back here and then I'm very happy to share my lesson with you today. Okay, so let me share screen. All right. If you have any questions, please do ask. Yeah, I'll try to answer your questions. Okay, so let's start with this. Okay, chapter 3 is one of the most important topics of Form 4. Okay, aside from all the other chapters about the physiology of the human body and so on. So this is the basic thing about biology that you need to know. What is how the plasma membrane is constructed. Okay, what are the structures in the plasma membrane that allows movements of substances into and out the cell. Okay, so let's start now. This is the general, the chapter, the name of the chapter, right? Structure of plasma membrane. So today I'm going to do 3.1, which is a short lesson. It is very easy, okay? It's just something basic for you to understand and remember the structure of the plasma membrane. I will not do 3.2 because 3.2 is quite a long subtopic. So it will take too long a time if I combine both together. Okay, let's do 3.1. Now let's look at this cell. All right, now this is a cell, this is an animal cell. Now, why do we need to have substances moving in and out of the cell? Okay, so we come to the problem of the necessity, the question of necessity. That means why is it necessary for substances to move in and out from the cell? Okay, so firstly, let me ask you a question. What does a cell need in order to continue living? Okay, so as you know, if all of us, right, humans, right, we need to eat, all right, we have to have nutrition. We also need to get oxygen, all right. So these are the two of the basic things that we need to supply to the cell: nutrients and also oxygen to be supplied to the cell so that the cell will be able to carry out an important process called respiration, or we call it cellular respiration, or we say internal respiration. Okay, so I don't see anyone with me on the channel today, but I know there are some of you here. If you would like to let me know who you are, just type say hello and ask me a question or anything so I can make contact with you. All right, so it doesn't matter. All right, if you're there, so very good, you're there, and just listen to the lesson. Okay, so these are the things that the cell needs to in order to survive. Okay, at the same time, as you know, cellular respiration, it also produces some waste products. Okay, so when the glucose is oxidized with oxygen, it produces things that the cell does not need. This is called the waste products. Waste products are like carbon dioxide. Okay, so for nutrients and oxygen, they need to diffuse. Diffuse is just a fancy word for saying that movement of molecules. So molecules are moving. Okay, when you say diffusing, means they are moving. So diffuse into means moving into the cell. Okay, so at the same time, it also produces substances that the cell does not want. Okay, from the metabolic process, all right, of respiration and so on, it produces carbon dioxide and also urea. So these are the things that we consider them as waste products. So they have to be removed from the cell. Otherwise, if it's not removed, it will become poison. It will become toxin that it will harm the cell. Okay, so these substances need to move out. So substances need to diffuse out from the cell. Okay, so that it may continue. The cell will be continue to carry on living, all right, and carry out its process. Okay, so you notice that the only way it can move in and out is through a very thin membrane here, which you have learned in Form 1. This is called the cell membrane, or to be specific, all right, we call it plasma membrane. Okay, so this is the what they call the membrane that separates the cell from the external environment, from all the surrounding that is outside, all the material and substances that is outside the cell. Okay, so that is the plasma membrane. So you should in Form 4 here, you should use the term plasma membrane instead of cell membrane. Okay, plasma membrane is the type of membrane. Okay, cell membrane just refers to the membrane that covers the cell. Okay, so we need to be specific here. So we call it plasma membrane. All right, okay. So let's look at the plasma membrane today. Right, now, all cells require substances from the external environment, that means outside the cell, so that the cell can carry out the living processes, respiration, okay, producing energy, and so on. So at the same time, metabolic processes, like things that the product of respiration, like carbon dioxide, which is a waste product, it needs to be removed from the cell. Okay, so it has to move out. So the only place to move out is through the plasma membrane. All right, so this cell must be able to allow the substances to move in and out, so it can get what the cell wants, and the cell can also remove what the cell does not want. All right, okay. So this movement is regulated or controlled by the plasma membrane. And plasma membrane, you will see in many pictures, right, later on, that it looks like this. Okay, so if you were to magnify it, all right, we are not able to see all the structure actually, because they are very, 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 very small. Okay, so it's just a model that some scientists have proposed, and they said that it's all, it looks more or less something like this. Okay, so you'll find that whenever you see this structure with a lot of these blobs embedded into this two double layer here, that looks like a plasma membrane. Okay, so now we're going to look at the components that make up this plasma membrane. All right, okay, so there's no comment yet. All right, let me carry on now. So the structure of plasma membrane. So this structure, of course, many other scientists earlier have proposed their model, right? But this is the one that we still use until today. This is called the fluid mosaic model, which was proposed by S.J. Singer and G.L. Nicholson in 1972. So this has been found to be most accurate to explain, okay, the movement of substances into and out of the cell. So we still use this model until now. Okay, so basically this model looks like this. Okay, this is a picture I've taken out from our textbook. So this one shows very clearly what are the parts that you need to know and know how to label. Okay, very important that you know how to label it, because it is very likely it will come up in a question. Okay, what is this X? What is Y? So you have to know how to recognize and label it. Okay, so you will see there are many, many parts here. So we'll go through it one by one. All right. Now, so it's made up, basically, you find that it's a layer. All right. Actually, you have this molecule here that looks like a little round ball here with two tails, right? So the entire molecule here is known as a phospholipid molecule. Okay, which has got two parts. So you will see here, right? There are two parts. The one on top here is called a head. All right, a head means kepala, right? So this part here is called the head. Sorry, let me go back a little bit. So this is the head, all right, which has a certain property. Okay, this property is known as hydrophilic. Okay, after I explain on the next slide, I'll explain what's the meaning of hydrophilic. And then you have two tails here, jutting out here, which is known as hydrophobic. The characteristic, okay, or chili is considered hydrophobic. So you have a head with two tails, right? So this is one molecule of phospholipid, and you need many, many, many of it to form two layers. So we call it bilayer. You see, there's one layer here, right? And there's another layer here. You can see here. So the head is like facing outside, right? And with the tail meeting each other inside the internal part of the membrane. All right. So this is phospholipid bilayer. Now, not only that, you see inside this phospholipid bilayer, you have a lot of these blobs. All right. Berketul-ketul lah. Okay, all these things that is macam berketul-ketul. So this is like molecules. These are protein molecules. Okay, they are embedded. Okay, they are stuck inside there, right? Inside the phospholipid bilayer. Okay, but bear in mind, this protein molecules that are embedded inside, they are not fixed. They are not what they call the position of these molecules are not fixed. They are able to move around. Okay, so that means at one moment it looks like it's there, the next moment it can move to another place. So it's not fixed, it's a dynamic kind of sort of movement. So this membrane is known as a dynamic model. That means it changed. Okay, the pattern, the mosaic, the pattern here changes from time to time. So that's why we call it this word fluid. Okay, fluid means it's like liquid. It is able to move. So all these substances, all these things that are embedded there, they are not fixed position. All right, okay? So you have seen the phospholipid bilayer with the protein molecules. Okay, there are other things that you'll see. You'll see the glycolipid and the glycoprotein. Okay, that one later. Okay, let's go to what we want to tell you about the molecule, phospholipid bilayer. So you will see if I magnify this, all right, you will see that there's a head here. This is the entire phospholipid molecule. You know, I'm talking about this one only. Okay, actually you need the other one to form the layer. So it's called two layers, bilayer. So the head is actually called a polar head. Polar means it is attracted to water. Okay, it's like more or less something like charge. There's an idea of something like charge. Okay, so this is made of phosphate component, right? You will have your okay, let's see. This polar head is known as hydrophilic. Just now I mentioned this word hydrophilic. Hydro means water. Okay, whenever we say hydro is water, right? Philic, okay, philic is from the Latin word, actually Greek also. It means philos or philos. It actually means love. Okay, so you can see the L here, how to remember. Your philic is an L there, your love is L. That means it loves water. It likes water. It will be attracted to water. So this polar head will be attracted to water, or rather water will be able to move near to this and sort of like, attract the polar molecule. Okay, attract this head. So it's attracted to water and we also consider as water soluble. Okay, so the head is water soluble. Okay, then the other side where you have your two tails there, right? This is a non-polar tail. Non-polar means the opposite, that is not charged, all right? There's no unequal charges, there's no what we call electropositive or electronegative charges there. So they are it is considered as hydrophobic. Okay, hydro is again water. Phobic. Now, phobic is actually a fear. Okay, like phobia. Some people have phobia, right? They are phobia with what? Flying. They are irrational fear of flying, or they can't see blood. Right? When you see blood, they're gonna faint. When you see blood, they'll faint. Or some people are phobic with phobia with heights. They stand on top of a very high building and they look down, the knees will shake. Okay, that's phobia. It's called fear. So phobia actually from the Greek word phobos, which means hate, fear, or dislike. So this part of the non-polar tail is hydrophobic. It means it does not like water. It is not attracted to water. It is repelled by water. That means it is not soluble in water. If it's not soluble in water, that means it's soluble in the other thing, which is lipid. Okay, so most of the things here is either water soluble or lipid soluble. If it's not water soluble, it probably is lipid soluble. Okay, so it will dissolve in oil. Okay, if it doesn't dissolve in water, it might dissolve in oil. And then if the thing dissolves in water, it probably will not dissolve in oil. Okay, so hydrophobic, see the P here, H here, that means hate or do not like. So remember, philic is love, phobic means fear. Okay, it doesn't like water. So this is the characteristic of your phospholipid molecule. All right, okay. Now we need two of that, right, to like sort of sandwich it. All right, then you have your phospholipid bilayer. So here I'm going to show you the actual makeup of your phospholipid molecule. So you will see the head there. Okay, the head there is actually phosphate group. Okay, plus a glycerol there. So you see, it's all these atoms and molecules. They are very small. Okay, so we can only sort of like imagine. You can't see it, not even under the electron microscope. So each phospholipid molecule, right, consists of a polar head, which is hydrophilic, that means attracted to water. And then the this one here, that is the phosphate head, and the non-polar tail, which is hydrophobic, which repels water, which will move away from water. Okay, it's not water soluble. All right, this part here. So you got two. It's actually fatty acid. So all you see here is this carbon and hydrogen. The black one is carbon, the white one is hydrogen. So you have multiple CH3 all over the place, and it's very long. Okay, the chain is long. We call it a fatty acid chain. Okay, that's part of your phospholipid molecule, right? CH3. One black one is the C, carbon, attached to three atoms of hydrogen. Okay, either 2 or 3 lah. Okay, either 2 or 3. Okay, so the hydrophilic heads of the phospholipid molecule in the outer layer. So let's say there are two sides of the membrane. The one that show you the outside of the cell, this one is facing the outside. Okay, and this is the inside of the cell which faces the cytoplasm. So the hydrophilic heads, they will face the outer layer, that means the extracellular environment, and then the other head, right, will face the inner layer. So one head will face up here, the other head will be facing towards the inside. All right, okay. Let me write this first because after there's some animation coming up. Okay. Right, next one. So while the hydrophobic tails, okay, they are in between. They are on the inner side. Okay, the tails are facing each other in between. All right, okay. And this is how the structure of that's how we get this phospholipid bilayer, because you need two of them. Okay, the head facing out and another head facing the internal side of the cell. Okay, with the tails facing inside, facing each other. Okay, clear, right? Okay, so this is simple. We have just mentioned the phospholipid bilayer that makes up the backbone of the entire plasma membrane. But that's not the only thing. You have other things that is attached into your phospholipid bilayer. Okay, so the next one, let's look at other structures. Right now, there are other things called these protein molecules. They are embedded. Embedded means like stuck inside there. Okay, they are dispersed. They are spread all over, and it's inside the membrane. All right. Okay, so these protein molecules, all right, they have channels. Either there are two types. There are two types of these protein molecules. There's one type which has got channels. Channels, that means canals. Canals or channels. That means there is a we call it opening in the middle. Okay, this which allows things to pass through. So this is known as channel protein. Channel protein. And then the other type, which doesn't have a clear-cut kind of channel inside, okay, they are called carrier protein. So this one can change shape. The carrier protein will be able to change shape to bring in the substance. Whereas for the channel protein, it's clear-cut. There is a what they call a terusan. Okay, there is an opening inside there. So it's very easy for you to label. All right, if the question asks you to label, you always look at the diagram that has a pore that is always open. That is the channel protein. Okay. In last time, the syllabus, we use pore protein. The earlier syllabus, which is a KBSM, we use it a pore protein. Okay, but now in your new syllabus, KSSM, they use channel protein. So anytime you see the word pore protein or channel protein, they are referring to the same thing. Okay, it's just the term they changed it already. Okay, so this is your carrier proteins or channel proteins, or we call it transport proteins. They are embedded inside the phospholipid bilayer. Okay, now next one. So here is how you recognize if there ever is a question that requires you to label which is channel protein, which is the carrier protein. Look for the one which has got open. The pore is always open. If the pore is always open, right, that one must be the channel protein or pore protein. Okay, and then the other one, which is the carrier protein, the shape will be able to change. So it's not always the pore facing this side. It could be the other side, all right, and then the structure will change or turn over, and then the molecule will be able to be transported in or out of the cell. All right, so this is how you recognize that. So you see when the pore is always open, this one, this is the channel protein. All right, okay, next one. Now, other things, you will see there are some other things that are attached onto either the phospholipid bilayer or on the protein molecule. Okay, so they are called carbohydrate chains. So carbohydrate means you have this glucose molecule, and then it's all connected and connected. You get very long chain. This is a carbohydrate chain. They are either stuck onto this one, which is your protein, molecule, or it could be stuck onto the bilayer, the phospholipid bilayer. So there are two types. Okay, so the one that is stuck on the protein molecule is known as glycoprotein. Okay, glyco actually means carbohydrate. It's like sugar. Okay, carbohydrate is a type. Sugar is a type of carbohydrate. So glyco actually means sugar. So glycoprotein is this whole thing, all right. This whole thing is called glycoprotein because you have the protein molecule there, but you also see the uh, carbohydrate chain attached to it. So the entire thing is known as glycoprotein. Okay, the other one, you will see there's another type that's called glycolipid because you see it's attached onto the phospholipid bilayer. So that is attached to the lipid. So they call it glycolipid. All right, okay? So you need to know how to recognize which is a glycoprotein, which is a glycolipid. Okay, and then what's the function? All right, why do we need these things on the surface? Why is this carbohydrate chains attached to it? So they serve a function. Okay, one of the function here is they act as receptors to hormones. Okay, now so this is attached onto the outer side of the cell. So certain hormones, okay, when the body releases certain hormones, these hormones will act on the cell. Okay, the cell has to receive the hormone, and then this is the target cell. The cell will know what to do. Once the hormone chemical comes over, the cell will produce the response. So that needs a place for this hormone to attach. Okay, so this is the receptor. Receptor means is to receive. So when a hormone chemical comes, it will be stuck onto these glycoprotein structures. Okay, glycoprotein or glycolipid. So they are receptors to hormone molecules. Okay, once the hormone molecules are attached to these receptors, then your cell will produce the response that is needed. Okay, so they are receptors. Okay, then also to stabilize the membrane. Okay, by having these glycoprotein and glycolipid, okay, these chains, they will stabilize the membrane by forming hydrogen bonds with the water. So water will be there, right, and it forms hydrogen bond with the molecule of water to stabilize the membrane. All right, another one is to act as antigens for cell identification. Okay, they sound so difficult, right? Okay, let me break it down to you. Antigen is like a marker, right? So when this cell identification, we have this type of glycoprotein and glycolipid on top of it. It is like a marker for the cell. It's able to identify the cell. Okay, whatever substances that you have, the cell has some kind of identification. So every cell is different from each other. So you will have this kind of a chain attached to it that serve as an identification for the cell itself. Okay, antigen is like substances. It's like more or less most of it are proteins that attach to the outer surface of the membrane. So they function as an identification mark for the cell itself. Okay, so it's like just some kind of identification for the cell. All right. So these are the three functions of this glycoprotein and glycolipid. Okay, so this question came out before recently, just in our SPM question. Okay, so they asked you for the function of this glycoprotein or glycolipid. So you need to remember at least one of these points here. Okay, next. All right, now we have gone through, okay, so now there's another part here. We have gone through the phospholipid bilayer structure, also the protein molecule, then the glycoprotein and also the glycolipid. Now you will see in between the phospholipid bilayer, you will sometimes see some cholesterol molecules that are embedded, okay, in between the phospholipid tails there. So these are cholesterol molecules. So these molecules, the cell will produce itself. Okay, even we don't take a lot of cholesterol food, our body will also be able to generate or to produce or synthesize cholesterol. And they are needed actually. They are needed in the plasma membrane. Okay, cholesterol is needed there. Why? What is the function of cholesterol in the phospholipid bilayer? So it actually makes the phospholipid bilayer stronger. Okay, it strengthens it stronger, makes it more flexible. Just now remember we said the membrane is actually fluid. Okay, it's not rigid. It's not a hard thing. So it's more flexible because of the cholesterol and make it less permeable. So it doesn't allow many substances to go through. Doesn't allow the substance to go through here. Okay, like ions. water-soluble substances. It doesn't go through this this bilayer, but it will be able to go through this thing, the channel protein. Okay, so water-soluble substances, they do not actually pass through the phospholipid bilayer. That will be a second lesson. That is the 3.2. We'll talk about it next week. We'll talk about what are the things that can pass through the phospholipid bilayer. Okay, this phospholipid bilayer allows what to pass through. So one of the things that cannot pass through this phospholipid bilayer is water-soluble substances. So it's because of this cholesterol there. Okay, it is not water-soluble. So when it's non-water soluble, it will not allow water to pass through. Okay, so water, if it wants to pass through, it has to go through another entrance. The other entrance will be all these channel protein and carrier protein. Okay, so this is something that you're going to learn in the next lesson. So it's to make it less permeable. So water-soluble substances will not be able to pass through the phospholipid bilayer because of the cholesterol presence, the presence of cholesterol there. Okay, because cholesterol is lipid. So anything lipid will not allow water-soluble substances to pass through. All right, okay? So next. So again, I want to stress this to you. The phospholipid bilayer, the proteins and cholesterol, they are not static. Static means they are not movable. Static means they are not moving. So they are not fixed. They are able to move around. Okay, 1 minute they are there, the next minute they jump to another place, right? That's what it means. It can change position. This channel protein, this carrier proteins and all that, they are able to move around in the phospholipid bilayer. Okay, so they form a dynamic, that means it's always changing. Dynamic is something that's always changing, it's not fixed, it's flexible structure. So the structure is able to, move around. So after this, I'm going to show a animation to show that they are not static. All right, okay, so this contributes to the fluid characteristic of the plasma membrane. That's why we call it fluid. This is called the fluid mosaic model. Okay, remember these three magic names, the words are fluid because they are not fixed, right? They're able to move around. Mosaic is like a pattern. This one embedded here. It's like the jigsaw puzzle kind of thing. It's a mosaic, right? And it's a model. Right? We don't actually see it, but it's a model that is postulated by Singer and Nicholson. Okay, next one, yeah? I'm going to show you, this is an animation, right? You see that it's able to move around, so you can see the fluidity of the plasma membrane. Okay, this one doesn't show the yeah, this is the plasma. This one, this is the protein. The protein that is there. It doesn't show it moving around, but it's able to actually change position. Okay, so it's a fluid mosaic model. All right. Okay. So next. Okay, we're coming to the end of 3.1. All right. Okay, as long as you remember back all what I've gone through, all the different components and the function, and you must know how to label. Okay, now let's talk about this word, permeability. Okay, it must be something new to you. I'm sure you haven't seen this before. You have maybe heard of the word permeable, okay, but you may not really know what it means. So if I say permeable, that means if I say something is permeable to a substance, okay, it means the substance is able to pass through it. Permeable means it allows the thing to pass through. Okay, for example, my skin is permeable to, let's say, not permeable to water. Water does not soak into my skin, so it's not permeable. If it's permeable, it means it allows water to go inside. Okay, that's what it means by permeable. Allowing passage of substances is called permeable. Okay, so impermeable means it does not allow the thing to go through. Okay, impermeable. So if you put a IM there, is the negative of the original word. So you will see here, all right, you see that this membrane here, okay, if it is a selectively permeable membrane, that means certain things can pass through, but not other things. So you can see this membrane, the selectively permeable membrane, is allowing passage of the red color substances. Okay, these molecules that happen to be smaller. And the blue ones, you see, they cannot pass through, right? The blue one is like stopped, okay, by this membrane. So this membrane that are able to allow the substance, the red color, to pass through and does not allow the blue color thing to pass through, the molecules, we call it selectively permeable membrane. Okay, it does not allow everything to pass through. It is able to select. It is able to control. what can go in and what cannot go in. Okay, and that is the feature of the plasma membrane. Our plasma membrane has this ability. It can allow certain substances to pass through and certain substances cannot pass through. Okay, so our plasma membrane is known as a selectively permeable membrane. Okay, last time we used the word semi-permeable. Okay, but we will not use the old term. Now we use the new term because it's slightly different meaning. Okay, so we will follow the one in the textbook. They say selectively permeable membrane. All right, so it allows, definition, question asks you, what does it mean by selectively permeable membrane? It means this membrane allows free movement of specific molecules, certain molecules only across it can allow it to go through, and it will prevent or it will limit the movement of other substances. Okay, so usually it's based on the size. Okay, because sometimes these molecules have pores, means the membrane has got pores that has got specific sizes. So big molecules usually cannot pass through, the small one will be able to pass through. Okay, and also sometimes we can talk about the water soluble or lipid soluble. Example, just now I mentioned the lipid, okay, the phospholipid bilayer. It is because of lipid, water cannot pass through. So it's like it will be stopping water molecule substances, soluble substances to pass through the lipid phospholipid bilayer. But it will go through another way. Okay, it cannot come through this way, it will go through another way. All right, okay. So the phospholipid bilayer and protein molecules will determine the membrane permeability towards specific substances. So whether the substance can or cannot pass through is based on these two things, the phospholipid bilayer and also the protein molecules. So it's their characteristics. Okay, so certain things can pass through phospholipid bilayer, and if it doesn't pass through there, it has to pass through the protein molecules. So this is what the phospholipid bilayer and the protein molecules do. It is able to control, right, what can pass through, what cannot pass through, and that determines what can go through and what the cell needs and what the cell needs to move away, throw away or move out of the cell. Okay, all right. So you need to remember the definition of permeability or selectively permeable membrane, allowing certain substances to pass through, molecules to pass through, while limiting or preventing other molecules. All right. Okay. So that is the 3.1 lesson. It's short one. Okay, it's just the beginning. The next lesson will be more challenging, where you need to remember what are the things that can pass through the phospholipid bilayer and what are the things that will pass through the channel protein, and another one is which are the things that can pass through the carrier protein, and also the different types of transport. Okay, passive transport and also active transport. Okay, this is very important. You will find it in your questions, definitely. Okay, at least the question, every exam you have this kind of questions. Okay, all right. So I don't have any questions from you. So I hope that the lesson is clear. So please, you can share this link to anyone who your friends who may need the lesson. So if you have any questions, I will just let you know my Telegram, what they call that, username. So you can message me, and I will be happy to answer your questions if I can. All right. Okay. So I hope to see you next week. So so long from me. And I hope you have a wonderful weekend. All right. Thank you. And see you in my next lesson. Okay. Bye-bye. See you. Bye, everyone.